Hello friends, I am Antika and today in this video I will discuss about Ectocarpus, a brown algae, and it is of much economic importance and it's famous mostly as a source of algin and fucoidin. These phycocolloids are used in a range of food and as well as cosmetic industries. Apart from this phycocolloid, many other secondary metabolites from Ectocarpus is used in both food and cosmetic industries. So this will be the first algae that I'll discuss in the series of brown algae. There will be many algae discussed in this brown algae series. Among the phaeophytes or the brown algae, Ectocarpus will be the first algae because it is mostly the simplest of all the brown algae that we have in our MSc syllabus. And in this video I will discuss about the morphology as well as the ecological significance of Ectocarpus. In the upcoming videos I will discuss about the asexual reproduction, the sexual reproduction and the life cycle of Ectocarpus. And I will be making another video about how Ectocarpus is used as a model organism in various type of lab experiments. It is a model organism. So that videos will follow this video, and of course I will be putting the links in the description box of all the algae that I am making. So today's the topic of discussion will be Ectocarpus, which is the simplest of all the brown algae. Now, before I go to Ectocarpus, I should be telling that brown algae or the phaeophytes, they have their characteristic. They have many important characteristics that differ from that of the green algae. Now, green algae, they have chlorophyll A and B, but the as their main pigment. But in the brown algae, apart from having chlorophyll A and chlorophyll C, they have abundance of beta carotenes and various type of xanthophylls like fucoxanthine, violaxanthine, diatoxanthine. And in fact, these abundance of carotenes and xanthophylls, they mask the color of chlorophyll. That is why they appear, the brown algae, they appear either olive green in color or golden brown or dark brown in color. And also another thing that distinguishes brown algae from the green algae is that the stored food material is laminarin and mannitol and sometimes fat droplets but not starch. Fucosum vesicle that is present in the cell of brown algae is also a very characteristic of brown algae. And also the cell wall of brown algae is different from that of green algae. As brown algae, they grow to great heights, so they suffer from desiccation. So the cell wall, apart from having cellulose, is also made up of alginates and fucin that help to protect the algae from desiccation as well as from the rough wave actions. And these are the main points which distinguishes brown algae from green algae. I have made many videos regarding the many green algae, and if you go to my channel, you can have this in the list, that is algae. So let's start the video. So Ectocarpus, it is a brown algae, and it is taxonomically, so it is in a division Phaeophycophyta and class Isogenierate, order Ectocarpalis and family Ectocarpaceae. Ectocarpus siliculosus is one of the most commonly studied algae, that is, it is very common in lab. Apart from Ectocarpus siliculosus, Ectocarpus fasciculatus is also very common. In fact, this Ectocarpus, it is used as a model organism to study the mechanism, both metabolism as well as genetic machinery of other brown algae. Just because Ectocarpus is very simple in structure and it has a very small genome size, and so it is used both in as a genetic model organism in genetic studies, as well as model organism to understand the cell structure as well as the developmental stages in other brown algae. And a total 410 species of Ectocarpus is found in the world, that is the global. The species are found globally, and in India we have only 16 species in India. So this is mainly because this Ectocarpus, it prefers growing in cold area in the tropical temperate region. It is abundant in Atlantic, the temperate region of Atlantic, but also some areas in Pacific also. So Ectocarpus coniferous, Ectocarpus indicus, Ectocarpus arabicus, and Ectocarpus darmonimatus. These are the Indian species. So Ectocarpus, though it prefers temperate region, but during the summer season, if there is mixing of water, upwelling of nutrient, or if there is eutrophication, then we can see blooms of Ectocarpus. And this is not a single bloom, but this bloom includes both Ectocarpus and another algae, Pileila. So inhabited, it is mostly marine, and few brackish species can also be found to be growing on the river mouth in the estuarine area, that is Ectocarpus subulatus. And in cold area, it is found to be growing along with a red algae, Gracilaria. Ectocarpus, brown algae Ectocarpus is found to be growing along with red algae, Gracilaria. So they are found at growing attached to any rocky substratum or coral reef, or they are found to be growing epiphytically on other brown algae, especially belonging to this order Fucalis and Laminarialis. In fact, Ectocarpus coniferous and V. articulatus, they have a mechanism, they have an enzyme called dehalogenase that is used to neutralize the halogenated compounds that both Fucus and Laminaria produce to deter other algae from growing epiphytically on them. But just because this Ectocarpus species, they have this dehalogenase enzyme, they neutralize the halogenated compounds, and that is present on the laminaria and fucus, and after neutralizing it, it can easily grow epiphytically on laminaria and fucus. So this they are found attached with the rocky substratum or substratum on the reef, and also epiphytically on fucus and laminaria. They can be grown epizoically from the fins of the fishes. That is, in animals, on the surface of animal, that is the fin of fishes, we can find Ectocarpus fasciculatus. The same species can grow endophytically, that is inside the tissue of Laminaria digitata. So this is the habitat of Ectocarpus. Now Ectocarpus, it is of two types. One is the annual species and another is the perennial species. in this Ectocarpus fasciculatus, it is annual, and Breviarticulatus, it is perennial. So it is highly branched and it is olive green to golden brown color, and in structure, both the gametophyte this life cycle is actually, it has an haploid and diploid generation. So this type of life cycle is called the haplodiplontic life cycle, and both the life cycle is either isomorphic, that is similar in structure, or slightly heteromorphic, that is having a slight difference in the structure of gametophyte and the sporophytic generation. So two type of ectocarpus are there depending upon the species. Some are isomorphic, some have heteromorphic alternation of generation. But this alternation of generation is dependent upon many external factors. Especially one of them is temperature, which is major factor. And in fact, if we manipulate the temperature, that is under lab grown condition, we can actually obtain the gametophytic generation, that is it reaches the sexual reproductive stage at even very short height. So this is how the life cycle can be affected by temperature. Now back to the structure, the most species of Ectocarpus, they are isomorphic, which means that both the sporophytic and gametophytic thalli, they appear similar but not same, because the gametophyte, that is the N, that is the haploid generation, it is much tufted in appearance. It has higher number of branches, but the sporophyte, that is the 2N generation, it has less number of generation and less number of branches, and due to this fewer branching, they appear much less tufted than the gametophytic generation. But again, the number of branching, it all depend upon and the shape, the size, it all depend upon external features, that is the chemical nature of the water around it, the temperature, the salinity. So all these determine the external morphology of the ectocarpus. So Ectocarpus, its thalli can be divided into two parts. That is, it is heterotrichous in nature, which means that it has got this prostate system, this horizontal prostate system, and another that is the erect system. So this one is the erect system and this one is the prostate system. The erect system, it can grow 30 centimeter in length, and the prostate system, it is branched, but branching is very sparse, and actually most of the time it develops into pseudoparenchymatous structure, unless it is suffering from any mutation, especially ectocarpus. So one mutant of ectocarpus, it has got a certain loss of gene, it undergoes loss of gene function, and due to this the entire prostate system is absent, and this mutant species of this mutant ectocarpus, it actually appears, it is floating in nature. So apart from that mutant population, ectocarpus generally have a prostate system which is irregularly branched or pseudoparenchymatous, and the cell division is due to equal symmetric division. And this prostate system is either attached to the substratum by a sticky substance, or they develop the rhizoid. And the growth in the prostate system, it is apical, whereas in the erect system, it is different. In the erect system, they have this the erect system is uniseriate and monocyphonomous, which means that polysiphonoas is like they have a central cell and many surrounding layers. They have a central cell and they have many surrounding layers. But here in Ectocarpus, such type of cortications can be limited to only the lower region, that is the older region of the plant. And the erect system, it is generally 30 centimeter in length and it is uniseriate and monosiphonoous. It is highly branched and the gametophyte is much more branched than that of the sporophyte, and this branching they appear beneath the septa, which means that say if this is the filament, the Ectocarpus body is filamentous and they have this filament have row of cells, so they have this row of cells, and so branching it appears beneath the septa. This is the septa. This is the septa, and branching appears beneath the septa. So this is how the branching is. So branching is always lateral because it appears beneath the septa, but however it can appear dichotomous because of the evection current. Due to the evection current, the lateral branches are much raised in position, and due to this they might appear as dichotomous. Now every branch end or end of the filament, they have they either they taper or just end to a point. They appear tapering, that is they appear tapering, or they just end in a point. But whether tapering or end to a point, the ends they do not have much chromatophores, so they appear hyaline. Also, the area is highly vacuolated, and sometimes they have this pseudo hair. Pseudo hair is a characteristic of Ectocarpus because only Ectocarpus have this pseudo hairs among all brown algae, because brown algae they have proper hair on the tip of the on the end cell, but here in Ectocarpus it is only having pseudo hairs. And this erect system, they arise from the prostate system by asymmetrical division, and the older region of the erect system, they develop rhizoids, and due to this formation of rhizoids, the lower region or the older portion of Ectocarpus, they appear corticated. Just like when we studied another green algae, that is the Nitella, where we observed that the entire body is e-corticated, but cortication was observed only in the lower part. Just like same in that happened in Nitella, this is also the same in case of Ectocarpus, where cortication is limited to the lower region only. So growth, it is in the erect system is diverse. In the erect system, the growth is diverse, that is it can be apical, it can be intercalary, that is intermediate position also cell divide to produce to increase in length, and it can also be trichothallic. So we observed that in prostate it was only apical growth. So the cells of Ectocarpus, they are rectangular or barrel shaped cells they have, and the cells, they have a outer cell wall that is made up of cellulose and pectin. The outer layer is made up of pectin, and the inner layer is made up of cellulose. Now this cell wall, which is of bilayered, the outer layer apart from pectin, it can contain other hydrocolloids, that is the alginates and fucons. Both alginate and fucons, they are sulfated polysaccharides. Now these sulfated polysaccharides like alginate and fucon, they help in protecting the brown brown algae Ectocarpus from desiccation. Due to this, they not only protect the cell from desiccation, but also it adds toughness to the cell wall and protect it from very rough and choppy action of the waves. Both alginate are actually the salts of alginic acid, and so these alginates, which are salt of alginic acid, they give a very mucilaginous appearance to that of the ectocarpus. So apart from having this mucilaginous nature, protecting it from desiccation, fucan and alginates, they also have got anti-adhesive property which protect ectocarpus from acting as a host plant to other type of epiphytic algae. So this was all about the cell structure, the no, about the cell, the wall, the cell wall. Then we have this plasma membrane very close to that of the cell wall, and the plasma membrane, the membrane of the endoplasmic reticulum, the membrane of the nuclear membrane, they are also in close association with that of the chromatophore membrane. So this is the plasma membrane. Then the cell is largely vacuolated. It has got a central nucleus, and it has got ribbon-shaped chromatophores, and the chromatophores actually they are either disc shaped or ribbon shaped, but the pigments are mainly carotenoids in large amount, though chlorophyll A and C also present. And to this ribbon shaped chromatophores, they have this pyrenoids. Now the pyrenoids, they appear to be projecting from the chromatophore, and pyrenoids, they have a pyrenoidal sac surrounding them. The pyrenoid have a pyrenoidal sac, and surrounding this pyrenoidal sac, there are also endoplasmic reticulum. So this endoplasmic reticulum is also present there, and apart from this living organelles, there are also non-living inclusions in the cell of the ectocarpus. Mostly, the vacuoles, they contain various type of polyphenols, and in fact, one of the most important type of vacuole that is present in the brown algae is the fucosan vesicles. The cell contains these fucosan vesicles, and in all brown algae and including ectocarpus, and they contain a special type of phenol called fucosan. So this fucosan containing vesicles, they are a characteristic feature of all the brown algae, and they are present in ectocarpus as well. So this is all about the cell structure, and earlier in earlier slides I have given the structure thallus. Ecological significance of Ectocarpus. Here I am going to discuss its ecological significance, how it is acting as a host to various type of animals, especially the bacterial species, various fungi, that is the chytrids, humicids, and how it is ecologically significant. Here we see that they are the food to marine invertebrate, food and shelter of many marine invertebrates like the crustaceans, the nematodes, the protists. This is not protist but protist, P-R-O-T-I. This will be I. So it is a shelter, a plant for crustaceans, nematodes as well as protists, and they are the chief accumulator of iodine. So earlier all the brown algae, they were good source of iodine, and so it is one of the accumulator of iodine which is of economic importance for human being, and also it is one of the it causes much economic loss to human being as it is a fouling algae. We must be aware that barnacles, they are the major foulant of sheep and goat, but in many algae, especially laminaria and ectocarpus, they are also very invasive, and they act as a foulant to in sheep and goats. However, ectocarpus can be controlled with a copper-based anti-foulants. And this copper-based antifoulant is also very effective against the bivalves also. We know that bivalves, they are the crustaceans that are very common type of foulant. So that copper-based foulant acts both on the bivalves as well as brown algae like Ectocarpus. So many type of bacteria grow periphytically on Ectocarpus. This Marinobacter, Rosibacter, Halomonas, and Turptobacter, Curina rosea, Agrococcus, Alternomonas, and Methylophaga. All these grow periphytically on Ectocarpus. And in fact, this bacteria, they are found to be growing in Ectocarpus not as a parasite but as a they have a mutualistic relation. Especially they release plant growth promoting factors. These bacteria, they have a mutualistic relation with this Ectocarpus. Just like when we find many species of fungi, they are growing inside the plant, especially pinus and other plant as mycorrhiza. And also this is the same case, that bacteria growing and inside Ectocarpus and releasing plant promoting factor. But in that of mycorrhiza, they actually help the plant to absorb minerals as well as water. But here this bacteria, they also have this mutualistic relation. They live inside the plant body, but in turn they release this growth promoting factors of Ectocarpus. Ectocarpus is host to various fungi like the chytrids, the omycetes, the hypochytrids, and these are the species that infect the various species of Ectocarpus. In fact, not only the Ectocarpus relation is limited to bacteria, fungi, or the invertebrates, but also it has got genomic integration with many virus. In fact, many viral genome are found to be incorporated in the genome of Ectocarpus. So it has got ecological interaction with this fungus also. And this Ectocarpus, in order to protect themselves from the UV radiation, it produces many flavonoids. And we know that human, they have been extracting various form of flavonoids from different plants as well as algae. So this flavonoid production, it is all by the Ectocarpus. It is encashed by the humans, so it is a much economically important fungi, as it is a source of flavonoids, it is a source of iodine, it is a source of many phytocolloids, especially algine and fucoidin. So this is how this Ectocarpus is of economic importance also. So with this I come to the end of first part of the video, and the next part I will be discussing about the asexual reproduction in Ectocarpus. And many such videos will follow, including the sexual reproduction of Ectocarpus, the life cycle of Ectocarpus, how Ectocarpus is used as a model for genetic study or morphological study or developmental study. So all these videos will be following this video. And in any case you want me to make a video in any topic, please do write it in the comment section, and how you like the video do write in the comment section. And if you like the video, do share it with your friends, and your subscription will help me, encourage me to make more such videos. Thank you for watching this video.